All right. Ready? I think we left off here. So this is kind of a relatively new push of thinking about how to grow crops by paying attention a little bit more to learning from natural ecosystems and seeing if we can apply some of those ideas into maximizing yield in crop systems. There's some issues with it. Probably one of the big drawbacks is that the way we do agriculture, largely here in America, is with monocultures. And it's because it's easy in the sense of using, all the machinery has been designed for monocultures. It's easy to harvest, um, easy to manage. And so you can understand why monocultures gained in popularity and have been pretty consistent. But there's been a push over the last decade or so of the idea of like permaculture ideas, where you're learning to work with intercropping ideas where you don't have a monoculture. You have a variety of plants. The idea of companion plants has been around for a long time, lots of centuries. But the idea of companion plants is finding good combinations of plants that actually help each other out. And in the end, you get better yields. The Native Americans were famous for that idea with the Three Sisters cropping system. And so the idea that you had the corn and the bean and the squash or pumpkin of some sort growing together. The corn was very upright, so the bean could twine around it and have the corn for the support. The bean plants were fixing nitrogen, and we know corn is a hog for nitrogen, so having that bean plant partner is awesome for the corn. And then with the squash, they grow more horizontally onto the ground. So they have these big leaves that cover up weeds and provide retention of moisture in that area. And so that was probably one of the first ideas that kind of went by the wayside with the advent of monoculture agriculture type practices. Yeah. So it's understandable why monoculture is so popular, but the idea that maybe we can learn something from natural ecosystems has been gaining attention. And so this is kind of a comparison between the two. And certainly in a natural ecosystem, you tend to have good diversity. And as we've been saying, it's a general thought process that when you have good diversity in a habitat or ecosystem, that usually means it's a healthy ecosystem. This idea often gets underappreciated, this idea of the root systems. And that's primarily because we don't see them, so they're easy to not think about. But this idea of having different types of plants with different root systems can actually be very beneficial. You don't, if you have some roots that are reaching deeper to get access to deeper water and minerals versus some that are more shallow and spread out, they find that that's a good combination. We'll talk a little bit more about that when we get into bioremediation and phytoremediation. We know that roots are leaky. So if you have a monoculture of, in this case, it looks like they drew corn. If you have just corn plants exuding exudates, that's like always having one thing for a meal day in and day out. Essentially it's very narrow in what will be exuded from that one plant species. If you have a mixture of plant species, then you have a mixture of exudates, and it's like getting a more well-rounded meal, a little bit more healthy for the microbes in the soil. And as we know, gotta have good microbes in the soil, good diversity of them, good populations to support the plant growth. So again, they're showing them microbial diversity is lacking or lower in a monoculture, which again makes sense if the bacteria and fungi in the soil are only hanging out with corn plants. That's just, narrow way of getting fed, really. And so there are some really nice ideas with this, but putting it into practice on a large scale remains unattainable to date. This idea of intercropping and having mixed crop species for food production, it's just the labor costs would skyrocket because you would have to do a lot of manual harvesting rather than machine harvesting unless someone comes up with new machines that are a little bit better. No, on the other hand, a lot of farming practices are getting into the high tech end of things. And so some of the costs are coming down in terms of irrigation. They actually have robots that go along and can assess disease now and take moisture readings. And so drones, the use of drones. I mean, so agriculture is embracing, a lot of big ag companies are embracing technology. And so maybe there could be a way eventually where they'll rethink the idea of monocultures, but right now, that's not the way of it. And so, but I just would like you to think about these ideas and think about some of the potential drawbacks of this and comparing that to natural ecosystems. And when you think about the idea of monocultures, from a pathogen perspective, if you are a pathogen, so if this is a cornfield and you are a fungus that causes, I'm not kidding, this is the actual name of the disease, corn smut disease. That's a real thing. Corn smut disease. If you got into this field and all you see is acres of corn, I mean, wow, you're a very lucky pathogen. You're going to be very successful. You can spread quite easily from plant to plant to plant, because if you've ever been through a cornfield, they're really closely spaced together, very easy to hop, so to speak, from plant to plant. And so once you get your foot in the door as a pathogen, it can be very easy to spread. Compare that to a natural ecosystem. It's kind of like herd immunity. If you have plant diversity and the corn smut, maybe let's imagine there's some corn plants in here, but not as many. And you have a lot of other plants. You can imagine where the corn smut pathogen would have a hard time hopping around from corn to corn. It's kind of like herd immunity. If you have a population of people vaccinated and some people are unvaccinated, the unvaccinated get protected by all the ones that are vaccinated. Maybe that's not the best analogy, but that's what it made me think of here. So, from a pathogen perspective, this has been a long-standing problem of how to deal with once a pathogen gets into the field, and hence all the different pesticides and fungicides and all of that have been used. So, it can be incredibly impactful and can alter, history. When you think about the Irish potato famine, that just swept through the potato fields in Ireland and caused massive loss of food, starvation, people immigrating. I mean, it altered history. So it's no small thing when you think about this way of growing and thinking about pathogens and how they can be successful under those conditions. Okay. One last thing I wanted to mention. Again, going back to my postdoc advisor, she's got a podcast where she's interviewed. If you wanted to listen to this, I'm not mandating this, but this is something, again, going back to the plant pathogens. The streptomyces are what I worked with when I worked with her. Those are the bacteria that are odd because they look like little petite fungi. They grow in a hyphal growth form, except they're much more petite than fungal hyphae, and they produce all kinds of antibiotics. You've taken antibiotics produced by them by now, I'm sure. And so I like her perspective here, the quote from here that, depending on the habitat, pathogens may or may not be pathogenic. And so when you take plant pathology classes, you tend to learn, this particular pathogen is 100% nasty. It's never doing anything good. And now they're realizing, and she says we have to take a more nuanced view of this idea that it's not always the case. If you go and think about mycorrhizal fungi, some scientists would argue that sometimes they're not really pathogenic, but they can be a little bit parasitic under certain conditions. And so again, you're kind of moving along this continuum or this spectrum of roles that they may play in different habitats. So, you know, she's of the perspective that if we better understand the microbes, both the good and the bad, and use that knowledge, we can actually make some progress with, in her field of study, it's crop production, looking at agricultural systems. So, so something to think about. It's not always, you know, all these microbes that cause disease doesn't mean they always do that. And so there's been many cases, in fact, where certain endophytes that we have been isolated from plants, they're like, wow, that's a plant pathogen. And that's how they've always known that it's fungus it's a pathogen. And yet when it's endophytic into certain plants, they're finding it's not harming the plant at all. It's actually helping. So that was another big kind of aha moment, that you don't always have them just playing out one role. They can, not all, but some of them have this ability to be on the spectrum of how they interact. Okay. Well, I got to stop talking there about plants and microbes. It's a sad day for me. I could go on and on and on, but that's not the name of this class. So I will continue on into the next topic, which is microbes and animals, which is also quite fascinating. Don't get me wrong. So, by the way, I am searching for like a review type paper that I'll post for reading on these topics that we've been covering in this slide set on microbes and plants. I just haven't found the one that I want to post yet. So to be continued on that, I'll let you know when I post it. Okay, now let's see. So I think the next slide set's posted on microbes and animals. Right? It's in there. It's in there. All right. So, okay. I like this quote by Carl Sagan. He's the one that was married to Lynn Margulis of the endosymbiosis theory. So he's a very famous astrophysicist, and was very successful in his career, and also a novelist. He wrote several nonfiction or fiction books as well. The movie Contact with Matthew McConaughey and, oh, I can see her face. I can't think of her name. Jodie Foster. The movie Contact, he wrote a book called that, and they based the movie off that. So anyways, must have been an interesting conversations they had over dinner when they were married. Anyways, isn't that cool? Here we have a beetle. This is that Nikon Small World competition where you can take pictures. And so we're going to look at a few examples of small animals, starting out with that and thinking about that, because for the longest time they were overlooked in terms of the microbial world that interact with microbes. That's the face of an ant. Doesn't that look like, it reminds me of that movie Alien? Doesn't it? Thing that pops out of the stomach and chases you all around. I don't know if all ants look like that. No, it's like, have a beer, relax. You're so angry. Yikes. And this one, I don't know. I think that looks cute. Kind of like silly almost, but I suppose if that was bigger than me, I might think twice about that. Bold jumping spider. Beautiful. I mean, really, if you think about the ornateness of their body, and now start thinking about all the microbes that are on them and in them. And just like with us, we have so many microbes in our gut that help us digest our foods, and they have the same thing going on. One of the first experiments that was done was looking at microbes in termite guts. And I remember when that study got published, and there's this big hubbub from the regular people when they found out that somebody looked at the microbial community of termite guts, and it was off of a grant. So grant money paid for that research. And they were so upset that taxpayer dollars were going toward this research. It was like this big hubbub for a while. It made it into the news. And they said, how ridiculous to study something like this. And, certainly that was a non-scientist that was expounding on that idea. They didn't realize that sometimes fantastic discoveries can be made with something like that. That seems, if you're not a scientist, like, what in the world are you doing? And yet very valuable information. And that was one of the earliest examples of, hey, we got microbes hanging out in, the guts of these animals and insects. And, now we spend millions upon millions of grant money dollars looking at all the microbes that hang out with us and benefit us. So not so silly anymore, but one of the most famous examples of microbes that interact with animals in a profoundly significant way is the leafcutter ants story. And they have fungi that they farm. So ants are really one of the first farmers on the planet. 50 million year old relationship, at least, if not older. And I'll show you a short video in a minute here, but this idea that you can see all these ants foraging in the tropical rainforests, and they climb up on trees and bushes, and they cut away leaf tissue, but they don't eat it. They just haul it back to their ant nest, and then they feed that to the fungi that they have in their fields, so to speak, that they're growing in their nest. And so they kind of chew it up and get it all nice and sloppy and mixed it with saliva, even feces there, because it's, they fertilize with their own feces. And then there's the fungi there that are taking that macerated leaf tissue and breaking it down. And so fungi, as we know, have wonderful enzymes that can break down even, lignin and cellulose and all of that. For getting that fungus dispersed, well, it's on the ant's body. So as the ant is moving around, it can get dispersed. They're very well taken care of by the ant. They are very well grown and cared for. And the ant will eat some of the fungus, but not all of it. They know how to keep the fungus garden going. And one of the other huge things that they noticed is that the ants are really good at keeping fungal competitors and fungal pathogens from destroying the fungal garden. And so this was kind of a head scratcher when they started looking at this more carefully, like, how in the world is this ant? It was basically a monoculture of this specific fungus that the ant was raising and growing. And why wouldn't there be other fungi in there or bacteria getting in there that would, just like we see in our agricultural fields, once a pathogen gets in there, it's usually pretty easy to have it disperse and kill off and overtake. And so this was something that they wanted to study a little bit more deeply, at least some people did. So here you see the ants with their leaves, and it's amazing. I mean, they'll cut these big leaf sections and haul them back. And the fungi, like I said, they break them down, and then the ant will eat some of that fungus for their own nutrition. So here's the nest where they're growing. So all that white fluff is the fungal garden. And you can see that there's many, many ants that are tending to the garden and keeping things going smoothly. And then you can see some of the green bits. So they have brought some leaves back and are busy feeding that to the fungus in their garden. And so when they started looking at this, what they did was they take some of this fungus garden and some of the ants back to the lab and set it up there. And they were just like watching what was going on here and noticing, yep, it's just this one fungal species that the ant is maintaining. And how is it doing that so successfully? So the name of the fungus, when they brought it back and identified it, it's the Escovopsis genus. That's the fungus. Oh, that's the pathogen. I'm sorry. That's the pathogen that they discovered in the garden. So when they took it back to the lab and started um, taking samples and streaking it out onto agar plates and looking, they could certainly grow the garden fungus, the one that was being grown by the ants. But some other pathogens started popping up on agar plates, and they're like, huh. So there are pathogens in there. It's just they don't seem to be taking over the garden in any way. They're being kept in check somehow. So then they did an experiment. They're like, what happens if we take the ants away? What's going to happen? And when they did that, the Ascovopsis pathogen just overwhelmed the garden. And so the conclusion, based on some of their experiments, is that the ants are necessary, that they are actively keeping those pathogens at bay in some manner. And they wanted to know more about that. So let me see if I can show the movie here, and I'm going to pause the recording because I don't. Okay, now I should be recording. Okay. So when Cameron was working on this and they saw all those filamentous type hyphal growth on the ant, they realized it was too small to be fungal hyphae. And so this is the group of bacteria I just mentioned, the actinomycetes. So the Streptomyces genus of bacteria fits into this group. And they have that hyphal growth form. And initially they zoomed in, they said, oh, they're probably Streptomyces. But when they started to do some DNA sequencing, they realized it wasn't just the Streptomyces, they were finding others as well. And so Pseudonocardia, Mycobacterium, Microbacterium, they all belong into the actinomycete group and they're related, okay? And so they're finding that there are mixtures. The Streptomyces are famous for producing antibiotics, but so are, when they looked at these other genera that I have listed here, which you don't have to memorize the genera other than actinomycetes. I'd like you to know that word, but you don't have to memorize these actual genera. So they think that there's kind of a mixture of microbes that are on the ant's body that are producing antibiotics to inhibit the pathogen. And I mean, that's a really good question. Like, why, after, all of this time, why hasn't the fungus, the pathogen, gained resistance? We know so much about antibiotic resistance, and, that's a big worry currently. But they think that there's enough of a cocktail mixture of antibiotic producers in the ant nest. And because the ants are continually foraging out, they're probably recruiting new types of bacterial species that can produce slightly different antibiotics. And so it's hard, it's kind of like a moving target. It's hard for the fungus to develop true resistance when they're faced with all of these different chemicals and trying to not, trying to become resistant to all of that they can't do it. And so that's the current thinking on that, is that you just are continually hitting the pathogen with slightly different microbes that are producing slightly different antibiotics or maybe very different antibiotics. And that's enough to keep the pathogen at bay. And maybe the ant is doing some other things of just its actions. Maybe it's eating some of the pathogen or actually weeding it out. There may be that going on too. They're looking at it. This is still something that they're continuing to follow up on. I kept reading on this topic in the published literature, and Cameron Curry's group found that there's yet another major player in this interaction, and they're black yeasts because they look black when you cultivate them and grow them on agar plates. And So this is complicating the story a little bit because they're also on the ant body. They hang out near the actinomycetes on the ant body. And they're finding that they're kind of taking advantage of the bacteria. They're gaining nutrients from the bacteria as the bacteria lives its life and does its excretions. The yeast can take that up. And what they're finding is that the yeast can be kind of a strain on the bacteria. And in some other published studies, it looks like the yeast is maybe not the best idea in terms of the bacteria seem to suffer a little bit by that in terms of their growth. And so the question is, are they harming the ability of the bacteria to such an extent that they're not also, that they're also kind of interfering with the antibiotic production and potentially keeping the pathogen at bay? And so that was published after the initial, after Cameron published his master's thesis and got that out into the journals. And so this is from one of the publications, and it's complicated, right? You got all these swooping lines between the players and how they interact with each other. The plus means it's a positive thing. So they're trying to show here, this is the bacteria that hangs out on the ant. They're showing that the yeast is benefiting from the presence of the bacteria, but the bacteria is looks like it's being harmed by the yeast. So it's a negative sign there. And then this is the actual good fungus that they're cultivating, and then the pathogen, the Asclepias. So I'm not going to get really too far into this other than to say that, they're further looking into it and realizing that there's other players in the game, that it's it makes sense, really. All habitats have usually many different microbes in that habitat. So it's not shocking, really, that there's other microbes there that are in significant population levels to be, you have to consider them into the interaction here. I don't know if you can make this out, but those are two of the scientists, and look how deep they are. That's an ant nest. Isn't that massive? So they took the top off and excavated down to the bottom of it. I mean, that's just amazing. I didn't know they got that big. So it's kind of a shame that they destroyed that one. I don't think there's any going back from that. But then from this paper, that was published, then they found that there's nitrogen fixers in the garden. And so these are bacteria hanging out fixing nitrogen. And so here's a way for both the ant and the fungal garden to get a nice source of nitrogen. So again, the more they look, the more they find, and the more complex it gets with better data and a deeper understanding. So they were looking at that idea, like, okay, what's going on with the nitrogen fixer? So that's the queen, apparently, where they are putting a black arrow. That's queen of the ant colony. And she's hanging out on top of the garden. Here is some of the data just showing that you do have in the fungus garden, you can see a huge difference in the presence of the acetylene reduction activity. When you run that test, what you're really the question you're really asking is the dinitrogenase enzyme present in that sample? And you can see, yes, the answer is definitely a yes in the fungal garden, and that's the enzyme that fixes nitrogen. And so that's kind of a proxy for if there's plenty of dinitrogenase there, yep, there's plenty of bacteria there doing nitrogen fixation. And then again, just percent nitrogen content in the different organisms. You can see ants with their exoskeleton body, they do carry a lot of nitrogen and need a lot of nitrogen. So then they kept looking at it and they found out there's even more going on there. So constant threat, the ants are under constant threat from fungal pathogens, from the Escovopsis, which use various attack strategies. And then they're talking about the microbes that hang out on the ant's exoskeleton. Underlying this, you have interactions of mutualistic and antagonistic organisms there. And then they started isolating all the chemicals. Pseudomocardia and Streptomyces are there, producing their own chemicals. The ant nest metabolome. So metabolome is the idea of all the metabolites. They essentially are trying to say, what are all the metabolites that have been made by organisms in this habitat? And so they had much more complexity to that data set when they looked at the metabolome, and they started isolating some of these chemicals. And they're again showing that there's all kinds of defensive bacteria that seem to be present more than what they thought. And the red means that they're inhibiting. And the green means that's a beneficial interaction. So the story goes on. And again, just showing some of the chemicals. So they're really getting into it, trying to find out what these chemicals are. And of course, there's another reason they want to understand this interaction, but some of these chemicals could be very useful for human medicine and human applications. So they're also looking at it from that perspective. That's a colony of Streptomyces bacteria that you see here. And you can see how it looks blue. That's not Photoshop. Some Streptomyces have that baby blue color like that. And then you can see kind of those blobs on the top. To my eye, they kind of look silverish. All the little blobs like that. Can you see that? Those are essentially liquid droplets that oozed out of the hyphae, and that's pretty much pure antibiotic right there. So I've seen that a lot when I isolate Streptomyces from soil samples and grow them on agar plates. And I used to wonder what some of those, and fungi do this too. I see that a lot with fungi. They'll ooze out this liquid, and then it starts to dehydrate a little bit with contact with air and kind of get like a crystalline form. And when you take that and isolate it and look at it chemically, it's usually pretty pure antibiotic. So it goes on and on and on. So 50 million years of interaction, and you got a lot of complexity. And so you can see a variety of different chemicals. So you can imagine if you're the fungal pathogen, it'd be hard to develop resistance to all these different chemicals, right? I mean, that'd be a lot of work. It'd be hard. And so that's one of the thinking, they're thinking that there's just so many different bacteria that they're finding now in the colony that are producing antifungals. So interesting story that continues to go on. And, it is kind of ironic that it was the ants who figured out antibiotic treatment. Well, before we did, we didn't really start using antibiotics till, well, there was a little bit of antibiotic use in World War I, but it really didn't come about until World War II. So 1930s, they used sulfa drugs during World War I, and I guess technically those could be antibiotic-like molecules, but antibiotics produced by bacteria and fungi didn't come about until World War II. So 50 million years ago, that was already a thing. We just were oblivious to it. So again, sometimes looking into things that on the face of it from a non-scientist point of view might be like, why are you doing that? I mean, a lot of good could come from this just from the human perspective, let alone just understanding such an incredibly complex, interesting interaction. All right, I think we got to stop there, right? We're out of time. So we'll continue on with microbe-animal interactions, and then eventually we'll get into microbe-human interactions as well.